I'm going to give you about a 30,000 foot overview of the Hawaiian Heteroptera biota and then one piece of it, the Nabidae. Just for context, the Hawaiian Islands lie in the northeastern Pacific. The chain's about 1,800 miles long. Uh, has a whole series of atolls that stretch out to the northwest. So if you laid it on the United States, it would essentially span the continental United States. So we're really a much bigger state than people think. We just don't have a lot of land in our state. Um, Most of the biota is concentrated down in the lower right-hand part of the slide in the main Hawaiian Islands. And really, most of the habitat is on the higher islands of Kauai, Oahu, Molokai, Lanai, Maui, and Hawaii Island, also known as the Big Island. So, most of my talk today will deal with the nabids occurring here because there's only a very few that are up in the northwesterns, and none of those are endemic. Um, the biota in Hawaii, there's about 400 named species right now. They're in all sorts of habitats, including marine. We have halibades. It's one of the few groups of single island endemic species up in the atolls of the Northwesterns. The Lygiids up there are locally endemic. Usinger sampled them way back in the 30s. Probably there's about 550 total species of Heteroptera in Hawaii, which would make it about 10% of the Hawaiian endemic insect biota. And here's just, a set of exemplar slides of habitats you've got, rushing streams and nice coasts and even mountains with snow on top of them because they go up to 13,800 feet. Um, the history of species descriptions in this biota is very episodic. It's largely centered around a few particular individuals. White and Blackburn were early early people who worked on this. Kirkcaldy came in and strewed a lot of chaos. Usinger was there in the 30s and 40s, did a lot of very good work. Peter Ashlock spent some time at the Bishop Museum, and then in more recent years, Adam Asquith, the late Wayne Gagne, and myself have contributed to the history of descriptions. If you want to look at this in a different way, you can see that our process of description has accelerated over time. So actually, we're actually getting at the taxonomic impediment here, and there's more to go. But you can see that it hasn't been one of these things where we've been just idle. We've been moving right along at describing the additional species that we know of. In terms of discovery, Perkins got there in the 1880s and 1890s. Of course, the place was relatively intact then, so everywhere you went, you found new stuff. There was a lot of ecological disturbance after that from sugarcane, from fire, from grazing. And of course, during World War II, very little work was done. And then the Bishop Museum, you had Wayne Gagne and other collectors who were working very hard in the 60s and 70s. And then finally, more recently, a collaboration between Cornell University and the Smithsonian when I was there, which we called Bug Strafe, uh, put a lot of additional collecting effort into the islands and was able to use helicopter assisted surveys and other things to get to very remote places and find lots of new things. So our recent rates of discovery have actually exceeded those of the initial collections in the 1890s when everything was relatively intact, which tells you, A, this isn't a system in collapse, and B, it's very rich. And so the harder you push on it, the more you find. And if we add in the species that we know now are in collections, but that have not yet been described, that pattern then is even more pronounced because the previous graph was just for the things that have names. And you can see here that at least in the 2000s, because that's really when Jim Liebherr and I were working very hard on this sort of stuff, we really racked up a lot of new species in a hurry. The described biota seems to be fairly intact based on collection records at museums. Essentially, 72% of the currently described species have been seen in the last 30 years or so, and 7% have been seen in the last 50. So it isn't like some other groups where birds in particular, where they haven't been seen since, say, like 1900 or 1910, and they've never come back again. It seems that the Heteroptera are, in fact, still fairly durable components of the insular biota here in Hawaii. The same pattern prevails for the total biota. These are things that we've got, but that we, haven't described it. If you add that into the descriptions, once again, 76% of all the known Heteroptera, described and undescribed, have been seen since 1990. So the nice thing is they're holding up, and that isn't true in all. Many of the holometabolic groups have a harder time of it because um, the larval stages or the pupal stages are vulnerable to introduced ants. But because Heteroptera are, hemimetamorphic, they can run away in their immature stages, so they seem to hold up better. So during the last 20 years, 30 years, there have been 190 new species of Hawaiian Heteroptera described, and that's nearly half the currently named biota. So this works out to an average of about 6.3 new species racking up per year. And we still have at least 110 species in hand awaiting description. So if you average that out and you say, well, can you keep up that rate of 6.3 a year? It'd take us about 17 years to work through the currently known native biota. Now we'll find some additional stuff after that, but this is not an overwhelming taxonomic impediment. So if we can keep some level of effort going on the biota, my sense is we'll have most of it described within the next 20 years. So that's a good thing. And recent monographs have shown that many presumably small genera of endemic Heteroptera in Hawaii are in fact quite species rich. So you're looking at catalogs and you're going, oh, okay, well, Serona, there were only a couple of species of those, or, Orthotylus or Nesiomyrus or Nabis. And what you really find out is that when somebody gets in there and does a monograph, you go from one species to 40 in Serona, right? When Asquith monographed them in 1994. You go from one species of Nesiomyrus to 50 of them when the late Gagne, monographed them in 1997. You go from 7 species of Orthotylus to 97 after I got done with a series of monographs, and there's way more Orthotylus still to go. So you're going to have, 150 potentially Orthotylus, and yet if you had looked at the catalog, at say in the year 2000, you would have essentially assumed, oh, this is a small group in Hawaii. Far from it. It's an enormous group in Hawaii. And then finally, Nabids, where there were 25 species described prior to 1915, and Zimmerman treated them in the Insects of Hawaii series. But since then, we've discovered 45 more. Okay, in fact, many of them quite recently, like in the 1990s. And so, although the monograph isn't done yet, I'm actually laying it out right now because the descriptions are done and the illustrations are done. And I just need to mock it up and get it into Bishop Museum Press, and this thing will be coming out. So this is where you're going to go with Nabids. And, Nabids, they're a small family worldwide. There's only about 500 species and 20 genera. But in the Hawaiian Islands, you've got 70 species of that 500. And that's 13% of the world biota. But Hawaii is only 0.003% of the world's landmass. So you've got a massive number of Nabids sitting in a place that's really dinky. Oh, and they're 13% of the Heteroptera biota. There's no other place where Nabids are 13% of any Heteroptera biota. So why? It's probably a compensatory diversification due to the relative paucity of native Regiviidae, because Regiviids are not common in Hawaii, and they're all emosines. That's the only subfamily that's there. They're often in very dark places like under logs or in caves. We have multiple cavernicola species. So in the absence of Regiviidae, Nabidae appear to have said, yep, we'll take that niche. Let's go for it. Okay, where did they come from? Well, there's a morphologically similar radiation in the Society Islands of various wing forms and body forms that appears to be derived from tropical Nabids based on various internal and external characters, and that has affinities to Australia. But the Hawaiian Nabids that look like the Society Island Nabids, the ones down in Tahiti and all, which I already monographed a few years ago, they don't have the character states that would indicate that they are, in fact, derived from tropical Nabids. Instead, they seem to be a monophyletic radiation sister to a Palearctic group called Reuteronabids. And the late Itzia Kerzner pointed this out earlier, that it appears that really the Hawaiian things probably came from the Eastern Palearctic. Kerzner considered all of the species referable to the subgenus Milu, which Kirkcaldy had erected, named for the Hawaiian god of the dead, of all things. But Milu shares a lot of character states with Reuteronabis, and at various times, Kirchner vacillated as to whether Milu and Reuteronabis were in fact the same thing, and whether Reuteronabis should be synonymized under the earlier name Milu. It's not currently that way, so Reuteronabis is a valid Eastern Palearctic group, and Milu is an endemic Hawaiian group, but they're pretty close. A plesiomorphic paramere shape is fairly typical in Tropiconabis, and it's got this sort of just semicircular form, but with an apical lamella that folds over at the tip. And that's important because most of the Hawaiian species do not have the apical lamella. In fact, really, there's one in high elevation in Haleakala up in the subalpine grasslands where it gets pretty cold. Actually, it snows up there. And it does have that. Even so, it's not clear that it's a derivative of Tropiconabis capsiformis. It might just simply be a retained plesiomorphy in that particular species. On the other hand, you've got another whole group here that's got a multi-lobate paramere shape. And this is the same distinguishing paramere shape that Reuteronabis possesses over in the Eastern Palearctic. So it looks like this might be potentially the plesiomorphic character shape that indicates the first set of colonists. These species that possess this, they occur in dry habitats, often at higher elevations. And as I noted, Kirchner vacillated as to whether these should actually just be lumped with Reuteronabis or not. If not, they're probably sister. But then it goes in all sorts of different directions. So the nice thing about the Hawaiian nabids is you really don't have to dissect them internally for the most part and inflate the Ediogus. You can simply look at the parameres that are hanging off this big box on the rear end, and they are quite distinctive. I mean, some of them, like Saiban, almost look like Oriental weapons. I mean, they're crazy. So you usually can get your nabid, you can pull it out. If you get a male, you just look at the side of it, and the paramere is just staring right at you, and you can often just sort them out right like that. However, the Ediogus offers some really good characters, and in the internal sclerotization of the Ediogus, sometimes you have things where the parameres are pretty close, the body form's pretty close, but you don't feel quite right about this. You start taking them apart, and you can see that these the internal Ediogus contains these sclerites that Kirchner called hooks and combs. And hooks would be like the big sclerites at the basal portions of these figures I've shown. Further out, more distally, you can see that they start to pick up, especially in the uppermost figure. You can see some projections coming off one of the curving sclerites. Those are what Kirchner called combs. The interesting thing is these things, when the Ediogus everts, they pop out. Now, this is an example off a specimen that was preserved and it popped out. And look at this, they evert out, and you've got all sorts of interesting things going on, which shows that the combs actually, in many cases, are often paired structures that interlock when they're at rest, and then they pop open like a pair of jaws. So who knows what's going on with all of this? But these are really good character systems. It's just that the average person doesn't necessarily want to go to all the work to, prepare things like this. And in most cases, the parameters will still get you an identification. The female structures can be okay. This is the Nubicola group, and you can see that you've got these tumescences that can form at the base of the ovipositor, and the orientation of the ovipositor itself in many species, sometimes it's sort of nearly horizontal when you view it laterally, and in other cases, it's angled up nearly vertically, and the whole form of the gonapophyses around it. These can be useful in case you only have a female specimen. The genitalia, also the internal genitalia, these are figures that Kirchner sent to me before he passed away. And it's interesting because all the Milu have paired parietal glands, a symmetrical vagina, a short common oviduct, which is also present in Reuteronabis, and a ventral sac that projects ahead of the oviducts. So in many cases, the paired parietal glands are not typical in Nabis, okay, because it's typical. Usually you have one, and here you've got two. So all of these are a good another set that seems to bind the group together, the Hawaiian radiation as an actual monophyletic radiation off of perhaps a single common ancestor. So there's three major groups that you can find when you're collecting, and they're pretty obvious based on wing form. There's Macropterus species, 24 of those. None of these have a basal sclerite in the male Aedeagus, and ocelli are always present. Then you have submacropterus species. They are the majority of the species in the islands. They always have a large basal sclerite in the male Aedeagus, and ocelli are nearly always absent except in one species where they're very small. And then finally, the Micropterus species, the wing pads are just reduced down to almost nothing. They have this sort of violin shape to the body. There is no basal sclerite in the male Aedeagus except for a couple of species from Kauai, which that would indicate that perhaps there's been convergent evolution up there of this body form. And then the ocelli are always absent in these as well. So how have we found them all? The Hawaiian Islands are a very rugged and ecologically complex place to do work. And so there's lots of places for navids to hide. This is Waimanu Valley on Hawaii Island up in the Kohala Mountains. But there's plenty of other valleys on other Hawaiian islands that look like this. So this is tough terrain to get around in, which is why we often need to use the helicopter to get into these areas and do survey. You have alpine zones with freezing temperatures and regular snowfall. Okay, there's no native navids up here. There are some unusual flightless endemic Ligeads that actually do not feed on seeds. They feed on frozen insects that melt out of snowbanks. So the Ligeads seem to have basically colonized this zone. Nobody else has really had a chance to get up there. In the far background on that shot below the snow cap on Mauna Loa, this is from the summit of Mauna Kea at the old Hawaiian shrines, you can see a small white streak. That's the Mauna Loa Observatory where the CO2 curve, the Keeling curve that measures global carbon dioxide is generated. Okay, below the alpine zone, we've got subalpine grasslands and shrublands. These are mostly inhabited by members of the blackburn-eye group that have the parame shape very similar to Palearctic Reuteronabis. Sort of makes sense because these are really temperate habitats, islands of temperate in a tropical archipelago. And so sometimes you get low shrublands that would look like something in the American West. In other cases, you get dry forests, like you can see on the lower slopes of, or the middle slopes of Mauna Kea. But once again, freezing temperatures are a regular occurrence here, and all the species in this area basically end up being subjected to winter every night. So the species in these habitats, they're macropterous. They're usually associated with specific host plant genera, many of which are more broadly distributed in the Pacific and in the temperate zones, Chenopodium, Sophora, Dodonea, and Dubaudia, for instance. But you can see there's sort of this is the suite of the things that are up there. They all look fairly similar in body form, but the parameters are different. And then you get into wet rainforests down below. And this is, of course, what everybody thinks of when they think of Hawaii. But these are pretty wet forests, like 400 inches, 10,000 millimeters of rainfall per year. The submacropterus and micropterus, actually, I should have said, species are concentrated in these habitats. So if you want to find the flightless stuff, this is where you go. And you wade through these very dense, upland, wet, super wet forests, and gradually you can discover these species. How do you do that? Well, here's an example. This is stuff that's on Maui, okay, for the most part. So you can see you have a bunch of submacropterus species, you have a bunch of micropterus species, and this is just an example of really just the island of Maui, and it's not all of Maui. But this is these are the sorts of body forms you see for the navids that are in the super wet forests. The way you get them largely is you go in there and you get a sheet and you put it in under mossy trunks or under various plants, and then you fog it with a small scale pyrethrin fog and you wait to see what falls off, because most of these navids that are in these habitats are living inside of moss, to be honest. And so they're not just crawling around on the plants. You actually have to force them out of very cryptic habitats inside these very, very mossy forest understories. If you get further down, you get dry and wet and dry. There's wet music forest, there's dry music forest. Music forest is a big continuum of forests, but they actually have their own suite of Nabis species. And these sorts of forests have been really vulnerable to grazing and fire. So they're not nearly as extensive as they used to be. But these are the sorts of species, for instance, you see in there. Once again, it tends to be Macropterus species. But for instance, really rare, Nabis robinae, which is up there in the upper left-hand corner, that is known from two specimens, both taken only from the same native ebony tree about 8 years apart near the rim of Makua Valley. The big Nabis kapulupulu down there in the lower right, we've got that from two specimens taken about 20 years apart at different places on sandalwood, on santalum. So some of these are just extremely localized, extremely, at this point, rare taxa that are in the mesic forest formations. And then finally, you've got a lot of dry and mesic shrubland in Hawaii at middle to low elevations that once again has been largely destroyed by plantation agriculture and grazing, etc., and fire. This is in Haleakala National Park at the Kaupo Gap, which that's a nice example of it. And there you tend to see these more widespread species that range around, and some of them are found clear up into the northwestern Hawaiian Islands and some of the higher islands like Nihoa. Nabis terrai down there in the lower left is nice because it's got a variety of color morphs. That's a species that was formerly widespread and is now really actually getting to be quite rare. Okay, so as we currently know it, we have one previously described tropical Nabis. It's a widespread species. It was long considered invasive, but I think it probably is indigenous. It's on every island all the way up to Midway. And then you have 72 endemic species in Milu, 25 previously described, 47 new to science, and all of these are endemic to the Are they going to last? That's a good question. We've got real problems with invasive species, wasps, ants, rats, deer. And for instance, Nabis terrai, that shrubland inhabitant, used to be pretty widespread on Stiphelia, but introduced yellow jackets got in on, of all things, Christmas trees, and they really like to nest in shrublands, and they really like to provision their nests with nabids. So terrai has gotten to be a very rare species, and largely due to, I think, invasive wasp predation. Fire, of course, does a number on all these habitats. And like, for instance, there was a mirid, an undescribed species of Opuna mirids that was on sandalwood, on santalum at Poli Maui. And I'm glad I collected it because the entire stand got destroyed and I haven't seen the species since. And then finally, host plant loss. We're drying out here, although, okay, 400 inches of rain a year is a lot, 10,000 millimeters. But except for those super wet areas, in areas that are less wet, we're drying out up on some of the higher elevation ridge tops in places like Oahu. And so as the host plants disappear, Kadua finis used to be called Hediotis terminalis in nearly all the literature. As that starts to go away, we're also losing the mirid species that were associated with these plants. And they've got nowhere to run. Once you pinch them off the highest summits where it used to be moist enough for them, they're not coming back, and neither are the mirids that used to be associated with them, or the nabids, or any other. So anyway, I do want to thank a whole lot of people who went into getting the data for this talk, because it wasn't just me. And of course, there's a big, long list of these folks, and you really can't do this work alone in terrain this challenging and under meteorological conditions that extreme without a lot of help from a whole lot of people. So thank you to those folks, and I'll take any questions.
Okay, thank you very much.